Okay, the first speaker is Vegard Edvardsen. Please, thank you.
So this is my phone. Do you hear me now? Okay. So hi, my name is Vegard Edvardsen. I'm a PhD student with the Artificial Intelligence group at the Department of Computer and Information Science at NTNU in Trondheim, Norway. So today I will show you how I've been using computational models of grid cells to perform navigational tasks in robots. So in my research, I'm interested in learning about what we know about how the brain might perform navigation, and then try to apply that to robots. And specifically, I'm interested in building artificial neural networks that can solve navigational tasks in robots. And these neural networks should then be biologically plausible and make use of the principles that we know from the brain. So one area of the brain that is particularly interesting in this regard is the hippocampus. The hippocampus is known to be involved in spatial processing through a wide variety of cell types found there. These neurons have names such as place cells, grid cells, head direction cells, border cells, and so on. So, for example, place cells are a natural candidate for a cell type to include in such a navigational system. So, place cells are neurons that will be active usually in only one area of a room. So what we see here is a top-down view of a room that might be being explored by a rat. And we see these four colored circles that are intended to illustrate areas of the room where these four different place cells might be active. And then say the rat is in the part of the room where the red place cell is active, and it wants to go to the area where the green place cell is active. So say it remembers that at the green place cell there is some reward. So how would the rat know in which direction it should go to reach the green place cell? Well, one possibility is to have the rat explore the entire room extensively and then learn all these associations between different pairs of place cells and the associated direction of movement. So in this way you can know that red to green, that's associated with a southeastern movement or minus 45 degrees. But the problem is that place cells depend on the particular environment that the rat is in. So if you move the rat over to a different room, then these firing maps of the place cells might change around completely. So in this example we see that the rat has moved to another room, room B, and then we see that the yellow and the blue place cell, they stopped firing completely. Some other place cells started firing, so we have a new purple and an orange place cell. And then the locations of the red and the green place cell also changed around. So to go from red to green in this new room, you can't no longer go to southeast, you have to go north. So this is called remapping. So we see that place cell activity alone is not sufficient if you want to be able to calculate distances and directions to arbitrary goal locations. But in the nearby area of the brain known as the entorhinal cortex, we find a possible solution in a type of cells known as grid cells. So whereas place cells are usually only active in one part of the room, grid cells are active in multiple locations. And these locations form a hexagonal pattern that extends indefinitely throughout space. So in the figure here we see the firing locations where this one single grid cell will be active. So we can sort of see the hexagonal pattern there. So this pattern then can be described by three parameters. First you have the scale parameter that describes how far distance there is from one location of activity to the next. Then we have a rotation, which describes how the pattern aligns compared with some axis of reference. And the phase, or the offset, if you will, which describes how the pattern is offset from some given point of reference. So if you record from multiple place cells at the same time, we see that these parameters are not independent between grid cells. Several grid cells will share the same scale and rotation of the patterns, but they will have different phases. So in this example here now, we see that we have a red, a green, and a yellow grid cell. They all have the same scale and rotation in their grid patterns, but they have different phases. these grid cells then are said to belong to the same grid module. Now in contrast to place cells, grid cells maintain these relationships when the rat is moved over to a different room. So we see here that the firing patterns have changed, but they have all rotated and offset in the same way. So this sort of solves the problem of remapping from place cells. However, we see that this repeating pattern of the grid cells introduces a new problem. So the pattern is repeating, and it repeats in hexagonal tiles that fill up the available space. So if you know that one grid cell is active, you're only able to tell your location within one of these tiles. You need some more information to tell you which of these specific tiles you're actually in. Now the solution in the brain is to have multiple of these modules and to have an increase in scale. So to illustrate this, here we first see the activity pattern from a grid cell from one module with a small scale. And we see that the pattern is ambiguous, so the rat doesn't yet know where it is, only from this information. But then we can add in the patterns from grid cells with increasing scales. And we see that as we add more and more of these, we resolve more and more of the ambiguity. And then finally now, the only place where these four overlap is in the middle there, so the rat is able to tell where it is. And I would also like to point out that even if this final large-scale grid cell might provide a very low-resolution picture of where the animal is, we are able to supply more information from the smaller-scale grid modules to increase the resolution. So you can sort of think of this as a positional numeral system. So when we have this in place, we can see that grid cells organized into a set of modules of increasing scale might provide a sort of generally applicable coordinate system in the brain. Now this does not necessarily mean that we should exclude place cells from our system, and in fact in the brain, place cells and grid cells are in close communication. So we might imagine that grid cells provide sets of coordinates to place cells, and that in return, place cells can recognize landmarks from sensory inputs and then use this to calibrate the grid cell's estimate of where the animal is. Now these cells are able to keep track of the animal's location even when it's completely dark. So this indicates that path integration might play a role in the system. So in other words, that the hippocampus updates its representation of the animal's location based on self-motion inputs that describe the speed and direction of movement. And path integration is also a key element of some of the computational models that have been proposed for grid cells, as we'll see in just a minute. So the task that I have considered for this project is then as follows. We have a simulated agent that performs a random walk in a two-dimensional open space. And then at the same time we have networks of grid cells that perform path integration in order to keep track of where the agent is. And then when a certain amount of time has elapsed, the objective for the agent is then to go back to the starting location. So it should go back to the cross there. And it should do this based only on information from the network of grid cells. And we want to do this with a neural network. So first I will now explain how attractor networks are being used as computational models for grid cells. And then I will explain my own part of the system that calculates the direction to the goal based on this information. So in attractor network models of grid cells, we imagine that each grid cell is assigned a row and a column index in a two-dimensional sheet of neurons. And each neuron in this sheet, for example, this one that we see highlighted here, is connected recurrently with every other neuron in the network. So all of these grid cells are fully connected to each other. And the weights of these connections are determined by the distance by which they're separated in the neural sheet. So we see in the figure on the right-hand side here, the function that decides these strengths based on the distance. And we see that neurons that are located close by to each other in the neural sheet will have weak connections between them, and likewise that neurons that are far apart in the sheet will have weak connections. However, neurons that are located in a band at a certain radius around the receiving neuron will send strong inhibitory inputs to the neuron. So the consequence of this setup is that the network activity will settle into certain attractor states when we simulate this. So we see an example here. So we see the two-dimensional neural sheet represented as a matrix. So we have 40 times 40 pixels in the matrix here, and each pixel shows the color-coded firing rate of one grid cell. And we see that this pattern here emerges by itself from random initial conditions in the network. So if you then imagine that this pattern repeats at the vertical and horizontal edges of the network, we can see that this is a hexagonal pattern of activity inside the network. What happens then is that this population pattern is made to shift around in the network in response to the self-motion inputs. So to show you this, on the right-hand side we have the current activity of all of the grid cells in the network. So this is similar to the matrix we saw on the previous slide. And then on the left-hand side here, we will see the trajectory of the agent as it performs this random walk. So pay attention to how the pattern in the population matrix consistently moves in the same direction as the agent. So when the animal moves south, this pattern shifts downwards and so on. So this means that over time, this hexagonal pattern in the population activity will become visible through the activities of individual cells. And this is why we can say that these cells behave as grid cells. So now the agent has stopped northwest of its starting location, and to compare with the original state of this population matrix from the beginning of the simulation, on the right-hand side here, we see that the pattern has shifted upward and leftward from its initial state. So we see that the network has performed path integration. And now this is the point where my part of the system comes into play. So the objective now is to use the information from the current state of the grid cells and compare this to the original state of the grid cells, and then use this to calculate the direction back home. So the job is essentially to compare these two matrices and then figure out in which direction we can find the shortest possible offset between these two patterns. And so to do that, I have introduced a new type of neuron to the system, which I call the phase offset detectors, which I will show you next. But first I just want to clarify some terms. So by current state, I then refer to the state of this matrix of the grid cell population at the current location of the animal. So this will be the result of the path integration process at any given point in time. And then by target state, I mean the corresponding state of this matrix that we expect when the agent is back at the goal location. And now we've chosen a scenario where the agent is supposed to go back to the starting location. The target state will then be a copy of how the current state looked like at the beginning of the simulation. So in this example here we see that the target state has shifted slightly to the right from the current state. So this means that the agent should move east to get to the target state. So in our system, we assume that there is some neuron that is explicitly pre-wired to detect this particular combination of current state and target state. So I have indicated this in the figure by this rightward pointing arrow, just to make the point that in our system there is some particular neuron that detects this specific combination. But then the target location might be somewhere else, for example to the northwest of the current location. So that's another situation that we will have to detect. So we will have to have a phase offset detector for that combination as well. Or maybe the target location is to the southwest, so we will need a phase offset detector for that as well. So in general, the target location might be in any given direction from the current location. So we need to instantiate these phase offset detectors for a range of possible directions around the current state. But now we assume that the current location of the animal is represented by activity in the center of the network. It might instead be represented by activity somewhere else, for example to the top right corner. So we need to instantiate a similar set of phase offset detector for that location. And there are many different possible current states. So we need to pick all possible current state locations from a rectangular grid. And for each of these, we pick possible target states in a circle around them. And for each of these combinations of current state and target state, we create a phase offset detector to specifically recognize that situation. And this can then be adjusted by some parameters we have in the system. So now that we've decided on which phase offset detectors we need to introduce to the system, we have to implement them through a neural network. So to implement this in a neuron, we have a situation shown here. So on the right-hand side, we have the phase offset detector neuron, and we see the parameters specified there. And we see that it receives inputs containing the current state on the top and the target state on the bottom. And each pixel in these two matrices then represents an upstream neuron that sends signals to the phase offset detector. And the problem is then to detect the synaptic weights for these connections. So first we only want the phase offset detector to be active when the current state is satisfied. So I have chosen to do this by inhibiting the neuron when there is current state activity in the wrong location. So in this matrix we see that each pixel shows the synaptic weight for the respective pixel from the current state matrix, and red then indicates inhibition. So with this setup the neuron will be strongly inhibited and thus current state is in the correct position. And then if current state is in the correct position, the neuron should fire whenever there is activity in the correct location in the target state matrix. So I chose to do this by having strong excitatory weights from the salient target state neurons. So here we see the excitation indicated with blue. So this quick description was just to say that the phase offset detector mechanism can be implemented by simple neurons. So this is then the final picture of the system. So recall that we need multiple modules of increasing scales in order to compensate for the repeating grid cell pattern. So we see in the middle of the figure here that we have these different modules running down the figure. And then on the left-hand side we see that we have self-motion inputs that go into path integration networks in each module. We also have a separate target state matrix in each module. And we have a separate set of phase offset detectors in each module. And then on the output end of the system, we have a set of motor output neurons that receive signals from all relevant phase offset detectors in all of these modules. And these then produce the final signal. So I will then demonstrate what the system looks like in action. What we see here is then similar to what we saw previously. On the left-hand side, we have a plot of the agent's trajectory, and on the right-hand side, we see that we now have 4 modules in the system. And for each module, we have the current state matrix on the top, which will show the path integrator result. And on the bottom, we have the target states. So I'll just press play here now. So see that the current states move away from the initial states. And I'll pause it here. So now the agent has performed the random walk, and it's starting its return back to the starting location. So based purely on the comparison of the top and the bottom matrices, the system will calculate the direction back to the starting location. So when I resume the video now, notice how the current state matrices gradually approach back to the target state matrices. Also, you might have noticed that the matrices to the left move more slowly than the ones to the right. And that's because they represent space at larger scales, so they update more slowly. So just to resume the video. And we see the matrices approach the target state, and then it stopped at the location quite close to the target location. And we have another video. First the random walk. And then when the line turns blue, it's returning back. And it found the target location. And the final example is this. And there it stopped. So the last thing I want to show you is what the effect is of using these multiple modules of increasing scale. So we might ask ourselves, what would this result be if we only use one module? Well, what we see here is the result from 500 different runs of the system where we only have one module. So these black dots are the stopping locations of the system. And we also see a histogram of these distances. So we see that there are a whole set of locations that the agent thinks is the real target location. So this is because we only have a module with a small scale, and the grid cell signal becomes ambiguous quite quickly. So instead of using this small scale, maybe we can use a larger scale module instead. Well, then we get this result. So we see that almost all of the runs do end up in a cluster centered on the actual target location, but we also see that we don't have a very good spatial resolution anymore. So these stopping locations are quite far scattered around the real target location. But when we use 4 modules, as I have done in these simulations, we see that almost all of them end up relatively close to the target location. So we seem to have gotten a quite good trade-off between long range and good resolution. So to summarize, we've seen that the outputs from an attractor network model of grid cell can be used by a network of simple rate-based neurons to calculate the direction back home. And we have seen that the ambiguity inherent in the repeating grid cell signal can be resolved by using multiple grid modules of increasing scale. And I would also like to point out this recent paper that was published online now in June. So in this paper they also discussed the topic of decoding the activity from grid cells. So this is very relevant to what I presented here today. So what's the future work for this project? I've not yet tested this system on a physical robot, so it will be very interesting to see how the system performs in the real world. And for example, it's very likely that noise in the motor control and so on will be an issue for path integration. So that it might be necessary to introduce some kind of sensory inputs to the system in order to correct the risk. Also, what are we doing so far? There hasn't been any obstacles to the agent. So it has only had to go in a straight line to reach the target location. So if you have a more complex environment, it might be necessary to perform some sort of pathfinding in order to reach the goal. For both of these extensions to the system, it would be natural to look at place cells as part of the solution, to sort of stay within this framework of the hippocampus from the brain. So finally, I want to thank